All right, let's finish up the microbes and agents list with the Archaea. So we're not going to go over too many details here just because there's not too much that's important for the Archaea, but things that aren't on the slideshow in general, right? The genome is going to be like you can think of it as the same as the bacterial genome, where you have, of course, no nucleus since it's a prokaryote, and then you have usually one circular chromosome, and then you can also have several plasmids that are within the cytoplasm as well. And I mean, the general morphology, right? You kind of have the same similar shapes that you see in bacteria, right? The bacillus or coccus morphology most of the time. Um, and yeah, that's pretty much it. And they're all archaea are non-pathogenic as well, even though they're present within our body. So let's get into the first one here, which is Pyrococcus furiosus. So this one is heterotrophic, which means, of course, it can't produce its own glucose or its own energy source. It needs to consume other like sugar sources available in the environment in order to acquire energy. It's also an obligate anaerobe, so it can't survive when there's oxygen. It needs to be in places where there's no oxygen. And it's also a hyperthermophile, so this is just adding more detail onto the thermophile, psychrophile stuff we discussed last lecture, right? So hyperthermophile is a specific thermophile that thrives at higher temperatures, so around 100 degrees Celsius or around the boiling point of water here. And it was originally isolated from geothermal ocean sediment, which means that it's also a marine archaean. And yeah, they're coccoid, as you can see, so they're cockish, pretty much cockish shaped, and they're flagellated here, as you can see on one end of the cell. So the big thing about these guys, the significance is that they have applications in biotechnology. For example, they're hyperthermophile, so you can imagine their enzymes are highly resistant to heat, so they won't denature even in high temperatures. And then remember we talked about the process PCR, right, or polymerase chain reaction. If you don't remember specifically, you can go review the Archaea lecture where I discussed it a little more in depth. But essentially, what we're trying to do is if we have a sample of DNA, we can keep using not an artificial, but like a DNA polymerase that we insert in the mixture in order to keep copying that strand of DNA over and over again, so that we can end up with like millions of copies, and then we can do some analysis once we have more of the DNA, because like one single little tiny strand, right, that's going to be pretty hard to analyze. So recall that in that process, right, we found that we could use something called Taq polymerase from Thermophilus aquaticus, which is a different archaean that's not mentioned here. But that one, right, if you recall, the PCR process involves heating, cooling, and doing that several times over and over in cycles. So we needed a polymerase that's resistant to denaturation even in high heat, so that we don't have to add polymerase again and again each time we heat the mixture and cool it back down. So we found that Taq polymerase was another thermostable DNA polymerase that we could use to expedite that process. But one thing about Taq polymerase is that it lacks what's called a proofreading mechanism. So essentially, like the DNA polymerase in our cells, for example, right? It's as it's going along and copying the DNA, right? It sometimes, right, you can make mistakes while you're copying the new strand. So the DNA polymerase within our cells can look back and check at the nucleotide that it just inserted, to check if it's the right one or not. And if it made a mistake, then it can take it out and then put the right one back in. So that increases the accuracy of the replication process a lot. But this Taq polymerase actually doesn't have this mechanism. So that means that when we do our PCR, right, we're going to get some amount of mistakes because there's no proofreading mechanism there. Whereas this polymerase, DNA polymerase that's found in Pyrococcus furiosus, this PFU polymerase, this one actually does have that mechanism. So people have started using this one instead of the Taq polymerase because it has a higher accuracy, and it'll give you copied samples that are more true to the original sample that you put in the mixture. Okay, next, Methanococcus species. So remember, SPP is species, so this is the genus, and then we have many species that are categorized within this group. So this is a group of, again, obligate anaerobes. So they survive in the presence of no oxygen, and they're also methanogens, which means, or methanogens, which means that they produce methane as a byproduct of their metabolism. So again, these are coccoid and flagellated, as you can see here. And then some are mesophilic. We didn't go over this term, but that just means like, this means normal. So meso is like middle, right? So they like middle conditions. They like normal conditions. While some are thermophilic, like the P. furiosus that we looked at. So their metabolism, which is called methanogenesis, this allows them essentially to use H2, right, hydrogen gas. They get electrons from H2 and then they use it to reduce. Remember, reduction means gain of electrons. So you're shoving the electrons from the H2 onto the CO2 here. So you're reducing CO2 and converting it essentially to methane, and then methane is produced, right? And then instead of H2, they can also use this compound called formate, which the formula is here, HCOOH, or CH2O2. And this can also be used as an electron source in order to reduce carbon dioxide into methane. And then some species can perform nitrogen fixation as well, where we're converting the diatomic nitrogen that's in the atmosphere into ammonia. And we kind of went over this process in a few lectures ago when we talked about the root nodules, right, and then the bacteria in the soil. So you can review that if you need. And then Methanococcus, it has potential applications for the environment because remember, right, it's taking in CO2 and is converting it to methane. Of course, methane isn't the best for the environment either, but these guys can kind of take the CO2 that's within the atmosphere and then once it's converted to methane, right, we can actually harness that methane. And then we can isolate it and use it as a fuel source as well. And that is one way that we can kind of, I guess, mitigate the effects of the greenhouse gases that have entered our atmosphere. Oh, and one more thing about all the Archaea, right? They replicate just like the bacteria. So they use binary fission, right, where they split in the middle and they produce two identical cells that are the same as the original mother cell.